Howdy, welcome back. We are continuing our journey through chapter 14. So this mini lesson is going to be talking about reservoirs and infection. How do we get the pathogens into our bodies to start the disease process? All right, so the first topic is going to be reservoirs. There's three major reservoirs, and a reservoir is where the pathogen typically hangs out. Could it be found in the environment? Is it found in the body? Is it found in animals? So those are our three major reservoirs. We have animals, human carriers, and non-living reservoirs. So let's take a look at some examples. So animal reservoirs are where there is a pathogen that can infect humans, but also animals, but doesn't always cause disease in the animals. They can, but they could just live in the animal, out in the wild, this organism that's living in an animal, and then once it gets into the human, it causes disease. So I have a few examples here of animal reservoir diseases. These are called zoonotic. So zoonotic diseases are the ones that can kind of move from animal to humans. So we have tapeworms, right? So tapeworms are definitely a zoonotic disease. They can live in animal hosts. And then if we were to eat contaminated food with these tapeworm eggs or cysts, then we would get tapeworms. Here we have Lyme disease. So Lyme disease hangs out in ticks. So the bacterium, it's a spirochete right here. And so it lives in the ticks. When the ticks bite their saliva as they are sucking our blood, this is a full tick, super grody. They can transfer the spirochete into our body, and that causes Lyme disease. This is rabies. So rabies is a viral disease, 100% fatal, with a very few exceptions in all of human history, like less than 20 have survived rabies after showing symptoms. So rabies is a zoonotic disease. If you've ever watched Old Yeller, you know what I'm talking about. But it actually causes rabies in the animals as well, where ticks don't get Lyme disease. But dogs and other mammals can get rabies and then pass it on to us. So animal reservoirs where the organism lives in the animal community and then can pass that on to humans. We call that zoonotic diseases, okay? Then we have human carriers, where the only place where the disease lives is inside humans. Sometimes humans can be symptomatic or asymptomatic. So we're dealing with that right now with COVID. There's such a wide variety of outcomes for infection with this particular virus, where you can have people who are infected and be asymptomatic, and yet they can still be contagious and shed their virus to other people. And the research is still up in the air as to whether it is zoonotic. I've seen some reports where like tigers in a zoo had it, or some pet animals have it. So it might likely be a zoonotic disease as well, but for the most part, this is a human transmitted disease, even though they can trace it back to pigs or bats or whatever. We had camels for the MERS, and we had pangolin for the SARS. So there could be a source, an animal source, that moves into the human population, but then humans are the only carriers of that disease. So we're still up in the air about what's going on with COVID. So this example that I showed here is a really famous human carrier called Typhoid Mary. You may have heard that name before. She was a kind of a cook back in, looks like in the late 1800s, early 1900s, where she would go around, and she was an asymptomatic carrier of typhoid fever. And so a lot of the families that she cooked for came down with this disease, and some of them actually died. And I think there's something where she had to go and kind of live on a quarantine island in New York. Seems like I just watched a documentary about her story. Very interesting. But yeah, so she was a very famous asymptomatic carrier. She didn't have any symptoms, and yet she was shedding the bacterium a lot in where she worked. So got to be careful about those human carriers. And then lastly, non-living reservoirs. So these are where the pathogen can live in places not another living thing. So I'm showing tetanus here with our rusty nail is kind of typically thought of when you have tetanus. It is Clostridium tetani. So we have our little bacillus-shaped organisms with the endospore, and tetanic contractions where this individual is suffering from tetanus infection. All of the muscles contract because that's what tetanus toxin does. I found a picture of a green T-bone steak. I thought that was kind of interesting. Meat, water, soil, these are all non-living reservoirs. Here is kind of a drawing depicting the cholera outbreak because it was the contaminated water from the wells at that Cambridge and Broad Street pump with John Snow as the father of epidemiology. We'll see that a little bit later. All of these would be good examples of non-living reservoirs where the virus or the bacteria lives in the environment and then can come into our bodies that way, not from another living thing, animal or human. All right, so those are reservoirs, animals, humans, and non-living locations. Now, that we've identified where the bacteria can hang out, or viruses, the pathogens, I can say, what are the ways that these pathogens can get into our body? Well, we have portals of entry. So there's lots of different ways, and sometimes they're specific for the organism. So for example, respiratory diseases typically are going to go into the body via the mucosa of your face, like COVID, like influenza, tuberculosis, all of these respiratory illnesses that affect your respiratory tract. And then digestive, right? But if you have a digestive illness, you have to ingest that into your mouth. Anytime you're going to break your skin, either by a cut or a scrape or an insect bite, or what's called parenteral is like a puncture, like a stab wound almost. You have your other mucosal openings, so vagina, penis, urethra, and anus. Those are also places where pathogens can enter the body. And then for women, there's a very small amount, but a certain percentage of pathogens can actually cross the placenta and infect the developing baby inside. So these are the ways that bacteria can get into our bodies to cause disease. Okay, and so now once we can see where the pathogens can enter the body, once they do get in to actually cause infection, they have to find their host tissue. It's just not like they're just going to make you sick just by being in your body. They have to do something to your cells to cause the signs and symptoms of illness. And almost always that's going to be some kind of adhesion or recognition between a surface ligand or antigen on the surface of your pathogen and some kind of receptor on the surface of the host cell. So here we have our host cell. So maybe this is your respiratory cell. And I know this is a bacteria, but it's just calling it a microbe, so we can call it the coronavirus. So we have certain receptors that are part of our respiratory tract. They're actually called the ACE2 receptor. They've identified this is where the coronavirus actually binds to. And on the surface, it has these spikes. So these are the glycoprotein spikes that are part of the envelope on the coronavirus. And so then what it does is it binds, it makes a recognition between the ligand and this ACE2 receptor that triggers the virus to go into our cells. Now, I don't know if it is the membrane fusion or the endocytosis pathway that we talked about in chapter 13, but whatever the mechanism, once coronavirus binds to your ACE2 receptors on your respiratory pathway cells, the virus gets into your body, starts replicating and killing those tissues, and then your body responds by making a whole bunch of mucus, which blocks your respiratory membrane, and it makes it really hard for you to do gas exchange. And so those are some of the more severe cases where people are having to go on ventilators and dying because they just can't breathe. They're in respiratory distress. One of those mechanisms of antimicrobials that we talked about in chapter 11 would be to try and block this, right? So blocking that adhesion. If we can block the coronavirus from binding to the ACE2 receptor, that is a good therapeutic medicine. And people are working on that. So vaccines, everybody's talking about vaccines, that's great. But we need a therapeutic as well. So we need prevention and treatment. So people are working on ways, looking at the mechanisms of how coronavirus binds to our ACE2 receptors, and is there a target that we can prevent the adhesion of the virus to our cells? Because if we do, then that prevents infection, which is what we're looking for. Okay. The last slide is more of a kind of like a joke. You may or may not know who Yanni is, depending on how old you are, I guess. But this is a little joke. Yanni was a musician in kind of the early 90s, I want to say. And and he was on YouTube. There wasn't even YouTube back then. PBS. So the story goes for this Yanni pandemic. In the mid-90s, Yanni adopted PBS surface glycoproteins to escape aesthetic immune detection. The new protein coat made his concerts appear to be part of the trusted community resource that is public television. It is estimated that over 500 million people were infected by his Live at the Acropolis special. We are currently developing a vaccine that targets Yanni's lesser mutable, highly conserved inner proteins to slow the spread of this pandemic. Just kind of a jab at the domination of PBS from Yanni in the mid-90s. So these are his PBS surface proteins, and they're trying to find a vaccine to maybe target some of these other glycoproteins. Right? Kind of funny. Okay, so that wraps up our video on reservoirs and portals of entry and how the bacteria and the viruses get into our body to cause infection. And then our next topic is going to be disease, the nature of disease, what is actually happening with these pathogens once they get into our bodies to make us sick. All right, so I'll see you next time. Bye.